Welcome back to Day 2 of Innovations in Disease Modeling. My name is Princess Senate, conference producer at Technology Networks, and your moderator for this session. And I am pleased to welcome our keynote speaker of today from the National Institutes of Health, Dr. Passley Hargrove-Grimes, who will be presenting a talk on development of tissue chips as in vitro tools for disease modeling and efficacy testing. So Passley is a program officer with a focus on translational science at NIH, and she currently manages the NIH-wide tissue chip for drug screening program. So in this role, she works to bridge the gap between basic scientific research and the development of new drugs by facilitating collaborations between researchers, government agencies, and pharmaceutical companies. So Passley has a strong background in scientific research herself, having studied retinal degenerative diseases during her PhD and postdoctoral fellowships. So please join me in welcoming Passley. So following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And with that out of the way, I will now hand over to Passley to begin her presentation.
Thank you so much, Princess. And thank you so much to the organizers of the Innovations in Disease Modeling Online Symposium for the opportunity to talk to you today about how we are funding the development of tissue chips as in vitro tools for disease modeling and efficacy testing. I'll start with a brief outline of my talk. I'm going to begin with an introduction to the National Center for Advancing Translational Sciences, or NCAT's mission and vision, as well as our mission and programs in the Office of Special Initiatives where I work. And then I'll talk to you about some translational gaps, namely the drug development dilemma and the way in which we might bridge those translational gaps using human tissue chip models. The meat of my talk will focus on tissue chip models of rare diseases, tissue chip models of addiction, pain, and opioid use disorder, tissue chip models for COVID-19 and other health emergencies, and how we're using tissue chip models in the precision medicine space and how we're addressing human diversity. I'll last talk about the path to adoption for tissue chips, the challenges that impede this uptake, and our future goals, as well as our current and future funding opportunities. So the National Center for Advancing Translational Sciences, or NCATS, was started in 2012 with a mission of turning basic research observations into dynamic health solutions using the principles of translational science. And our vision is very simple. We want to bring more treatments to all people or all patients more quickly. We are disease agnostic at NCATS, and so we really work from the tip to toe. We are interested in rare, common, Mendelian, complex, you name it. We're interested in it. I work in the Office of Special Initiatives with these incredible people at the top of your screen under the guidance of our director, Danilo Tagle, who was slated to give this talk today, but could not be here, and I'm happy to be here in his place. So our Office of Special Initiatives mission is to address translational problems with innovative solutions through the development and implementation of disruptive technologies that will hopefully result in a paradigm shift within the field. These are the programs and initiatives that our office is currently working on, and they are color-coded according to the technology that they represent. I will be talking to you today about the tissue chips or microphysiological systems for drug screening program. We have really developed this technology over time, and we've really demonstrated its robustness, reproducibility, and reliability. And we're now in the dissemination portion where we're focused on obtaining regulatory acceptance for tissue chips as drug development, bona fide drug development tools. Okay, so I want to first start by talking about the translational gap, the first translational gap, which is the lack of models in the drug development dilemma. So as you all know, animal testing has led to critical insights and medical advancements for humans. And animal models really benefit researchers in understanding how to treat and prevent various conditions. However, these traditional models are not always predictive of human pathophysiological response. And why do I say that? I say that because the development of drugs and therapeutics is extremely slow and costly. And in fact, it's projected that a single drug can take over 10 years and $2.6 billion to reach the market, and that includes the cost of failures. This is highlighted in this top figure likelihood of approval from phase 1. You can see in the blue bar for all indications, this is 9.6%. So this is less than 10% likelihood of approval for all indications from phase 1 to phase 2 clinical trials. So why are we having these issues? 55% of drugs are failing due to lack of efficacy. So they simply do not ameliorate disease condition. 28% are failing due to unexplained toxicity that were found in humans that was never found in the animal models and 2D cell culture models that were used to obtain the data to file that IND. So despite significant developments in computational biology and vitro biology and toxicology over the past 2 decades, only about 5 to 7% of human diseases have available therapies. And we think that's a problem, and that's a problem that we can hopefully try to solve. So in addition to the lack of relevant models, the therapeutic modalities that are currently existing on the market are increasingly human-specific and personalized. And so when I say therapeutic modalities, I'm talking about small molecules, cell therapies, gene therapies, ASOs, ADCs, and protein therapies. And so based on these 2 major translational gaps, the lack of relevant models and the fact that the therapeutic modalities are human-specific and very personalized, we think that there's a huge need for human-based model systems, and we're hopeful that this is where tissue chips will be able to come in. So tissue chips are also called microphysiological systems or organs on chips, and I'm going to kind of use those interchangeably within my talk. Tissue chips are these small bioengineered devices that are capable of recapitulating key functional aspects of organs and tissues. And they range from about the size of a USB thumb drive to multi-organ systems that can take up about a 96-well plate. They have 3 major features that separate them from other in vitro models, including a 3D tissue configuration, much like a normal human organ, multi-cell type composition, as well as the use of biomechanical forces. So for example, if we're going to create lung tissue, we're going to stretch that lung tissue biochemically with biochemical forces to mimic physiological breathing. So not only can we reconstitute the organ microarchitecture and tissue-tissue interfaces, but we can precisely control the fluidic environment. We can provide oxygen and spatiotemporal chemical gradients to the tissue chip. We can provide nutrition and removal of waste products. We've recently, to make these more physiologically relevant, have begun the addition of circulating adaptive and innate immune cells and features of the living microbiome, as well as incorporating inline sensors to monitor tissue viability and function over the course of weeks and months, which is how long we can keep these tissue chips alive. And so in order to not kill off the chip for endpoint analyses, we're able to use these inline sensors and be able to monitor the tissue viability and the function over time, which will really help us better understand human physiology in general. So the NIH Tissue Chip program started in 2012, and it really was a flagship program of the National Center for Advancing Translational Sciences. And we've tackled 3 major areas since our inception about 11 years ago. So in 2012, we really started with safety pharmacology, which was kind of the low-hanging fruit, asking ourselves if tissue chips really could mimic organ structure and function, and could they identify compounds that were found to be toxic in humans that were not found previously to be toxic in animals. And we were able to do that over the course of 5 years. In 2017, we moved into disease modeling and efficacy testing. And as I mentioned at the beginning of my talk, we are disease agnostic. And so we've really looked at modeling all types of diseases, rare, common, complex, Mendelian. And I'll talk to you about a few of those during the meat of my talk in just a moment. And then in 2020, we moved into the precision medicine space in our newest clinical trials on the CHIP initiative, and I'll be happy to talk about that in just a few moments. Okay, so for the meat of my talk, I'll start by talking about tissue chip models of rare diseases, and then I'll talk about a tissue chip model for addiction, pain, and opioid use disorder, as well as a tissue chip model modeling COVID-19 and associated pathologies. And lastly, how tissue chip models are being used within the precision medicine space to address human diversity. So 30 million Americans currently suffer from a rare disease, and many people with rare diseases first endure a very long and complicated journey to get an accurate diagnosis, with few, if any, treatments existing. And of the more than 7,000 known rare diseases, only about a few hundred have active research programs, which really leaves a significant gap in available treatments. And our goal is that tissue chips might help fill that gap. Okay, so I want to start out by talking about a 3D model for rare neuromuscular disorders. And this comes from a company in Orlando, Florida, Hesperos. They were one of the first human body on a chip companies. And they have developed a 3D human model mimicking the biology of chronic inflammatory demyelinating polyneuropathy, as well as multifocal motor neuropathy. These are rare autoimmune neuropathies that are characterized by muscle weakness, commonly resulting in difficulty ambulating and impaired function. In these diseases, the immune system actually makes proteins called antibodies that damage nerve cells and slow down messages moving from the brain to muscles. Current treatments are helpful, but often very inconsistent. So the researchers here developed a tissue chip model with two different cell types, motor neurons and Schwann cells. Motor neurons transmit messages from the brain to muscles, while Schwann cells help the signals move more quickly. The researchers showed that by exposing the cells to blood sera from people with these rare diseases causes a shower of autoantibodies from the immune system against the cells. And you can actually see this in the figure on the right. All of these arrows are actually pointing to autoantibody formation, not only on the Schwann cells, but on the motor neurons. And this leads the motor neuron signals, the signals to move much more slowly. So after treatment with a drug called TNT-005, they were able to block the immune system reaction and lead the message speed to return to normal. Now, excitingly, and the reason that I share this is because this is the first investigational new drug that has been filed using MPS efficacy data, leading to the authorization of a phase 2 clinical trial. And so the majority of the efficacy data for this IND came from tissue chips, this tissue chip that I'm sharing with you today. And that led to the authorization of a phase 2 clinical trial that is currently underway. I've put the link below, and as I know that these slides will be shared, and you can check out that link below as well. Okay, so in addition to the neuropathies that I just discussed, we also are developing a premature vascular aging on a chip model. Now, that doesn't sound like a rare disease, but what this is really modeling is Hutchinson-Gilford progeria syndrome. For those of you who don't know, progeria is a rare 1 in 18 million autosomal dominant, fatally progressive premature aging syndrome, whereby the patients exhibit exaggerated accelerated aging and cardiovascular disease and normally die from accelerated atherosclerosis around 14 or 15 years of age. The thickening of the adventia in the aorta is a hallmark of this disease, and you can see that to the right bottom histological figure that I have. So the project goal here is to develop a premature vascular aging on a chip model with iPSC-derived fibroblasts, smooth muscle cells, and endothelial cells derived from patients for drug screening. In the first few years, the teams were tasked with engineering a whole thermoplastic chip, which they did. And they're now in the second phase where they're developing 15 patient-specific chips with inline sensors and testing a variety of drugs to find additional therapeutics to treat disease pathology. These drugs include statins, biphosphonates, and farnesyltransferase inhibitors. Now, 15 patient-specific chips does not seem that impressive, but through working with the Progeria Research Foundation, which is a biobank as well as a patient advocacy group, this actually represents 6 to 7.5% of the global progeria population. So this is actually quite impressive, and we're really excited to see the outcome of this work within the next year or so. Okay, so not only can tissue chips be used to model rare diseases, but they can also be used to respond to national health emergencies. Before the global pandemic in 2019, the largest epidemic in our country, and still is one of the largest epidemics, is the opioid crisis. And so through the Helping to End Addiction Long-Term Initiative, or the HEAL initiative, we were able to issue 5 awards for tissue chips to model nociception addiction and overdose. And I'm excited to talk to you in a moment about one of those chips that we have developed in line with this mission. And then in 2019, our worlds were forever changed by the COVID-19 pandemic. And so through the CARES Act, tissue chip funding supplemental opportunities were rapidly issued to our awardees to develop unique tissue chip models for COVID-19, to understand multiple tissue and organ pathologies, to model infection, to test for candidate drugs and vaccines, to understand immune responses, and to model complications from vulnerable and at-risk patient groups. So I want to first start with a project that's focused on better understanding pain and the opioid use um, and opioid use disorders, and that comes from Hang Lin and his team at University of Pittsburgh, where they're generating an innervated knee joint on a chip to study osteoarthritis pain. So, as many of you know, especially those of us who have arthritis, pain is a predominant symptom of osteoarthritis, and radiographic changes are indirectly correlated with pain severity. Therefore, the source of the OA pain is not very clear. So, what researchers did here was they developed a multi-component miniature knee joint or mini joint on a chip using human cells. The novel design itself facilitates tissue communication while maintaining respective tissue phenotypes for over 2 months. The mini joint itself exhibits physiologically relevant changes when exposed to IL-1 beta mediated inflammation, which is very comparable to clinical observation. The mini joint can predict the in vivo efficacy of different drug treatments or the therapeutic effect of different disease modifying OA drugs. Here, they've taken their mini joint and they've innervated it with neurons from the dorsal root ganglia, which is the dorsal root of the spinal nerves, which carries messages from the peripheral to the central nervous system. And so what they're currently doing is assessing the impact of opioids, such as morphine, on neural activity in the presence and absence of joint injury triggered with different stimuli, as well as the integrity of all joint elements. So, in addition to this, we've also, as I said, funded work to develop tissue chips for COVID-19. And this comes from this work comes from Don Ingber's laboratory at the Wyss Institute at Harvard. I'm going to start by discussing the chip itself, which is all the way over here on the left. This is a human lung airway chip. It's an optically clear silicone rubber device that contains 2 parallel microchannels separated by an extracellular matrix-coated porous membrane lined by primary human lung airway epithelial cells cultured under an air-liquid interface on one side in the airway channel, with human pulmonary microvascular endothelial cells grown on the other side in the presence of continuous medium flow to mimic vascular perfusion. And they can add or take away human immune cells within the blood channel as well. So what they did here is they took these human lung chips and they infected them with pseudotyped severe acute respiratory syndrome coronavirus 2 or SARS-CoV-2, and then added clinically relevant doses of anti-malarial drugs and found that amidiaquin, which you can see in the purple arrow, inhibited infection, but that clinical doses of hydroxychloroquine and other antiviral drugs that actually were able to inhibit the entry of SARS-CoV-2 in cell lines under static conditions did not in their human lung chip. So they also show that amidiaquin shows substantial prophylactic and therapeutic activities in hamsters that have been challenged with SARS-CoV-2 on the right. The human airway on a chip actually may accelerate the identification of therapeutics and prophylactics with repurposing potential, and amidiaquin as a drug is now in clinical trials across 20 sites in Africa. So we've talked about tissue chips for rare diseases, tissue chips for opioid use disorder, and tissue chips for the COVID-19 pandemic. I now want to switch gears a little bit and talk about using tissue chips in the personalized medicine space in our newest clinical trials on the CHIP initiative, whereby we're trying to use tissue chips to inform clinical trial design, planning, and execution. We want to establish patient recruitment criteria. We want to stratify patients based on response or lack of response to a candidate therapeutics. And we want to develop clinically relevant biomarkers and reliable clinical trial endpoints. In the first phase, teams were tasked with developing and validating disease models with patient-derived cells. In most cases, these are stem cells or stem cell-derived materials, because although we know primary cells are much more mature both phenotypically and functionally, primary cells represent a very finite source of material that cannot be used kind of everlasting. And so we wanted a renewable resource, and that's why we really pushed the teams to use stem cell-derived materials. And then in the second phase, teams are currently working on testing potential drugs for efficacy and safety assessments in clinical trials. All teams that are funded within this initiative are directly comparing their tissue chip data to clinical data, whether that's retrospective, prospective, or alongside a current ongoing clinical trial. These are the teams that are currently being funded within the clinical trials on a chip initiative. And as you can see, they really are from tip to toe. We really have focused on not only common diseases, more like tendon inflammation and fibrosis, but also on rare diseases such as progeria, which I recently discussed. My apologies. So the first example of this clinical trials on a chip initiative project comes from Aram Han and Ram Kumar Menon, who are at UTMB in Texas. And they have developed one of the first fetal maternal interfaces on a chip to study preterm birth. Now, for those of you who don't know, preterm birth is considered live birth before 37 weeks of gestation, and it's a leading cause of neonatal morbidity and mortality, affecting 10.5% of pregnancies in the United States each year. The exact cause of preterm labor resulting in preterm birth is very unclear, but it is most likely a final outcome from activating one or more biochemical pathways. It's thus now considered a multifactorial syndrome with heterogeneity in both pathophysiology and biomarkers. And we really will need a better understanding of risk factors, risk-associated pathophysiology, biochemical markers, and mechanistic factors that activate the premature labor process to better understand and predict preterm birth and develop interventions to prevent preterm birth. So I first want to talk about the fetal maternal interface model itself. You can see this over here to the left. It's composed of 4 concentric rings, collagen chambers that are designed to mimic the thickness and the cell density of the fetal maternal interface in vivo. And it includes cells, primary cells from the decidua, chorion, and amnion, both mesenchyme and epithelium. Each layer is connected by arrays of type 4 collagen microchannels to recreate the basement membrane of the amniochorion. And they have shown that the cellular characteristics, such as cellular transitions, migrations, morphology in this tissue chip are similar to those seen in utero, and really validating the physiological relevance and utility of this fetal maternal interface on a chip. They created an ascending affection model through exposure of the decidual or maternal side to LPS, and they show that the LPS is able to propagate all the way to the amnion epithelial cells within about 72 hours. They've also developed an oxidative stress preterm birth model using cigarette smoke extract, and they've also used the model to predict responses to environmental toxins, such as cadmium. In this current project, the fetal maternal interface will be used to study the impact of racial and ethnic diversity, as well as the sex of the fetus on preterm birth. They'll be performing a prospective clinical trial and testing exosome-based therapeutics that have been shown to be efficacious at preventing and delaying preterm birth in mouse models. And so they're going to work now to see if they can reproduce those results in this fetal maternal interface on a chip. So the next project I want to discuss comes from David Beebe and Joshua Michael Lang at University of Wisconsin, and they're studying mechanisms of microenvironment-mediated resistance to cancer cell service targeted therapeutics. They're focusing on a major public health problem, which is metastasized advanced castrate-resistant prostate cancer, which is invariably lethal with no curative therapies. Their goal here is to engineer a tissue chip model of the bone marrow microenvironment to identify castrate-resistant prostate cancer therapeutics. In the first phase, they optimize the chip, which you can see here to the right itself. It includes a prostate cancer spheroid here in pink, surrounded by bone stromal cells in yellow, and this includes osteoblasts, osteoclasts, adipocytes, and fibroblasts. M2 macrophages in red, and lined by derived endothelial lined microvessels here. And so they demonstrated normal, healthy, as well as disease chip environments that accurately replicated in vivo physiology. And they've now moved on to phase 2. They're basically essentially using clinical trial data to build tissue chips that represent responders and non-responders to a candidate ADC that's currently in phase 2 clinical trials. They're taking tumor biopsies and blood from patients for molecular analysis, including RNA sequencing. And then they're analyzing that data and identifying signatures associated with relevant tumor microenvironment subtypes, such as M2 macrophages and osteoblasts, to build chips that represent tumor microenvironment of therapy responders versus non-responders. These signatures can be used to stratify patients for more efficient clinical trials. But we know that the path towards precision medicine involves modeling population diversity within these projects and all of our projects, and most likely using induced pluripotent stem cells that come from the patients themselves. So Jonathan Himmelfarb at ICANN School of Medicine and Matthias Kretzler at University of Michigan are developing kidney MPS models to model rare and common kidney diseases with a focus on African American minorities who are genetically vulnerable due to APOL1 risk alleles. The Pooh, Parker, and Bezerites lab at Harvard are developing cardiac MPS models to predict therapeutic responses to catecholaminergic polymorphic ventricular tachycardia patients. And they're actually recruiting protected populations such as children, neonates, and pregnant individuals that are currently not able to enroll in clinical trials based on protected status. Okay, so that gives you kind of a flavor of and some examples of the different tissue chip models that we have funded for development. And I want to kind of end my talk, or have this last kind of part of my talk be on how tissue chips, how we're accelerating the path of adoption of tissue chips, the challenges that are impeding the uptake, as well as the future goals, and our current and future funding opportunities. So the goal for us has always been to increase confidence and adoption of this technology, which is rather young. It's only about 15 to 20 years old. We started in 2012 with physiological validation. So asking ourselves, can we prove, can we demonstrate that we can capture organ structure and function in these chips? And do they respond accordingly to known reference compounds that, as I said before, were shown to be toxic in humans, but were never shown to be toxic in animal models? And can we recapitulate those findings? And in fact, we were able to do that. Our developers published over 500 original and review articles, many in top-tier journals, including Nature Medicine, Nature Communications, Nature Materials, PNAS, and Science. 5 years later, we moved into analytical validation, where we funded independent tissue chip testing centers, MIT and Texas A&M University, to onboard our developers' technology and to test them independently for robustness, reproducibility, reliability, and relevance. This is using a validation set of compounds, biomarkers, and assays. At the same time, we funded the University of Pittsburgh Microphysiological Systems, or MPS database, to take all of this data that we had from our developers, as well as our tissue chip testing centers, and have a general global repository for this data. In 2019, we moved into industrial validation, where our goal really was that these tissue chips would be used by industry and regulatory agencies. We know that they are being used in this capacity now with proprietary sets of compounds, and our tissue chip testing centers became commercially viable on their own without NCAT support, and evolved into Javelin Biotech and Texas A&M Tissue Chip Testing Consortium, respectively. And our University of Pittsburgh MPS database evolved into Biostatistics Analytics platform. And the Biostatistics Analytics platform has since merged with a major tissue chip company called Nordis to become Pneuma Biosciences. And so we see a lot of growth in this area, and this is extremely exciting for us to see. So we're here now. Okay, so we have a lot of challenges that are impeding uptake of this technology. The first is that these devices simply aren't standardized, and we do need some standardization to be able to move forward. We also don't have enough detailed use cases for these tissue chips to replace or support existing drug discovery assays, and we're going to need more examples of that for the field to grow. The cost of implementation is also extremely high. And there is a requirement for specialist skills and training to be able to run these devices. And so we do need these devices to become more easily workable, if you might say, and more turnkey, so that you don't have to be a bioengineer to be able to run these types of experiments. And then we have a few areas of improvement. We need the technology to mature. We need more robust validation of the translational and predictive in vivo-like biology. And we need quality standards for commercial viability. And so we have 4 major goals in order to kind of complete, to hopefully kind of upend some of these challenges. The first is we want to globally harmonize the regulatory use and standardization of these platforms. And because of that, we have created an MPS World Summit. This is the third year that we're funding, our third and final year that we're funding the MPS World Summit. We really hope that if you're interested in tissue chip technology or microphysiological systems, that you'll consider joining us here. It will be June 10th through the 14th in Seattle in the Convention Center, and you can scan the QR code to learn more or visit us online. Our second goal is to train the next generation of MPS scientists, and we're doing this through an international MPS society that was started about a year ago. And we also want to qualify MPS as bona fide drug development tools, and we are doing this through our translational centers for microphysiological systems. These, we have 3 awardees for this that were just awarded a couple months ago, and this will be a 5-year program with hopefully 5 qualified MPS, after the 5 years. And so the teams that were awarded here have taken 4 to 5 validated MPS with highly specific contexts of use. And when I say context of use, I mean, are the tissue chips going to be used for safety pharmacology or tox? Are they going to be used for disease modeling and efficacy testing, or are they going to be used within a precision medicine context? And that's just a couple contexts of use, and there are a variety of others. They'll be working hand in hand with the Food and Drug Administration to qualify the MPS as a bona fide drug development tool with a highly specific context of use. And then once qualified, these MPS will be made publicly and commercially available. And then the last kind of future, what's going on in the future for us is this complementary program or complementing animal research and experimentation. This comes from the Common Fund data ecosystem. And the purpose here is to catalyze the development, standardization, validation, and use of human-based new approach methodologies that will transform the way we do basic translational and clinical sciences. There are 4 major goals here. The first is to better model and understand human health and disease outcomes across diverse populations. As I alluded to before in my talk, we really need to work to incorporate diversity into all the experiments that we do. Tissue chips are one way, and new approach methodologies are one way that we can better understand diverse populations. Number 2, we want to develop new approach methodologies that provide insight into how specific biological processes or disease states occur. We want to validate mature new approach methodologies to support regulatory use and standardization. And as I said, we are already doing that in our translational centers for MPS, where we're working to take validated MPS and work hand in hand with the FDA to be able to qualify these for very specific contexts of use. And also complementing or replacing existent traditional models and making animal research more efficient and effective. If you want to learn more about our complementary initiative, you can do so I have the link at the bottom of my screen, and those funding opportunity announcements should be coming out later this year. And so this is my final slide, where I really want to thank my program director, Danilo Tagle, for the work that he's done in this area and how he's really accelerated this field forward. My fellow program officer, Dimitri Krepke, and our program analyst, Chris Sundrick. And last but not least, I want to thank all of our partners that we've continuously worked with over the past 11 years in this area, including the FDA, the EPA, NASA, BARDA, ISSNL, CASIS. Our IQ Consortium MPS affiliate, which are a large group of pharmaceutical companies that have come together to talk about which standards and which assays and experiments, et cetera, should be used for these devices, particularly within the context of the pharmaceutical industry. And I also want to thank our Trans NIH MPS working group. This is over 55 different program officers from a variety of NIH institutes and centers. We meet monthly to go over progress, delays, obstacles, and ways in which we can partner and collaborate better together to achieve our end goal. And so with that, I just want to say thank you so much, and I'm happy to answer any questions. Thank you.
Right. Thank you so much, Passley, for that fantastic talk. As Passley already mentioned, we are now moving on to the Q&A segment of this session. So we have received a number of questions submitted earlier on via our registration form, and I can see we still have a lot of incoming from our audience right now. So to all our current viewers, if you do see a comment or a question that you that was already submitted that you like, please give it a thumbs up, and I'll do my best to bring up the most liked comments or questions to Passley with the time we have left. So to kick um, yeah. So to kick things off, our first question delves into the application of this technology. So, Passley, you mentioned disease modeling in the context of rare diseases. Can you elaborate on how tissue chips are being used to model these complex conditions?
Sorry, I unmuted, but then it muted me again. So to understand your question, are there other rare diseases that are being modeled? Yes, there definitely are. Off the top of my head, we are modeling endometriosis, which is considered more of a rare disease, but definitely affects a huge population of individuals within the United States. So we've done that. I'm trying to think. I wanted to highlight the two major, but there have to be more. I can definitely come up with more, and I can put that on offline. I can connect with that individual.
Yeah, thank you, Passley. Yeah, like she's mentioned, we will be coming back with all of these questions via email as well. So yeah, so we did receive a fantastic question quite early on during your presentation, and the viewer kind of like raised a really interesting question about the tissue technology. So compared to traditional animal models, what are some of the biggest challenges and advantages of using tissue chips for drug discovery, especially for diseases with limited clinical trial opportunities?
Yes. So you make a really important point here. There are a lot of patients who enroll in clinical trials who, for whatever reason, are not able to enroll in subsequent clinical trials. And so what this would really allow is for this idea of tissue chips as patient surrogates. And so, for example, if we had a pregnant individual who needed to be treated for preterm birth, and we didn't know, which therapy would be the most efficacious, we could actually take fibroblast, make them into iPSCs, make them into different cell types in the fetal maternal interface, for example, and then test therapies for this individual tissue chip. But the question really was, what are the major advantages and limitations to using tissue chips versus using animal models? Is that correct?
Yes, that's correct.
Okay.
I think it's about.
So animal models, of course, are much more physiologically complex, and they really have been the gold standard for decades. Which and I highlighted the reasons behind that. One of the major issues is the cost of these animal models and the fact that a lot of them are not able to actually recapitulate kind of the complexity of the disease itself. So for example, one example I can give you is like for autism research, for example, we have not been able to, from what I know, create a very, very good model of an autistic animal. And so hopefully things like tissue chips could actually come in to be able to create a better model that is more physiologically relevant. The major issue now with tissue chips is just how difficult some of them are to run. You do need that advanced training. You do need, in a lot of cases, to be a bioengineer to be able to run these, and so it takes away some of the ability for other people to use them if you don't have that advanced training. But and I mean, another issue with tissue chips is that we can't recapitulate every single functional output, right? We can only recapitulate the key functional aspects and outputs of the disease. So there are going to be things that we're missing in tissue chips that we might be able to better answer in an animal model. I do like to mention that I think that we will achieve the best progress if we combine what we've learned from animal models and tissue chips, and if we use them both together. So I'm not advocating at all for not using animal models. I'm just advocating for using animal models more efficiently.
Fantastic. What a great answer. Yeah, so thank you for clarifying that. So our next question is about data sharing. So as a fellow former scientist, we all know data is essential for scientific progress. So following up on your previous question, actually your first question, so where is the data that's collected from these tissue chip studies deposited? Is there a centralized repository or database that researchers can access?
Absolutely. Yes, there is. This is the MPS beta database, which is now Numa Biosciences, which was Biostatistics Analytics platform. And I'm happy to share that link afterwards. This is a database that you can view all the data that has been published from these different experiments. You can view our developers' data, but you can also view the Independent Tissue Chip Testing Center and their data. So when they onboarded the developers' devices, you can see that data as well, and how reproducible it actually was. There are a lot of tools within the database now to analyze your data, whether that be from image analysis all the way to ELISA analysis. There's really, they're creating new computational tools all the time to be able to analyze and further kind of unpack your data. So happy to send out that link, and we would love, more and more users if possible, because this is, from what we know, the largest centralized repository of MPS data that's available.
Definitely, yeah, fantastic. Thank you. Yeah, so validation is key for ensuring reliability within these models. So, do you have any of these tissue chips been validated for specific diseases at all? Can you walk us through the validation process within the NIH tissue chip program for anyone that is interested?
Okay, so yeah, the word validation for us is really, have you published on the model? Has the model not only been published on, but has it perhaps been through additional labs through tech transfer? So have you taken the model and you've obtained the data, you've sent it to another lab to see if they have the same data or reproducible data? And so the validation network that I showed you, or the validation framework I showed you, really is what we have done. We really started with physiological, like we asked ourselves, can these devices really represent organ structure and function? And then we went on to let me just pull up my notes, my apologies. I want the actual note I have here. Okay, sorry. And then we moved on to analytical validation. And so we really used the tissue chip testing centers as a way to validate the data that had come from our developers earlier on. And now that data is still being validated by those independent tissue chip testing centers. They're just now commercially viable. And so we don't have any purview over them, but we know that they're still doing that work. And they're still working hard. We are now in the industrial validation portion. And so we have, hundreds of different tissue chip models that have been validated, but we don't have, what we really need is that those to go through kind of a qualification pipeline. We need the FDA to be able to take the data that was obtained from those tissue chip models and be able to evaluate its rigor. And so what we know now is that our pharmaceutical partners in general have a lot of MPS data that they're sitting on, and they've been using MPS internally for internal decision making, internal preclinical decision making for quite some time. What we're really trying to push for now is that they share that data either through the MPS database or through other kind of other sources, so that data doesn't just kind of sit there, and it actually kind of can go forth to help us better understand, this tissue chip technology itself, if that makes sense.
Fantastic. I hope that has answered our viewers' question. Let's just pivot a little to a different topic now. So, a viewer has actually raised a question about the broader applicability of these tissue chip models. So, beyond drug discovery, do you see tissue chips playing a role in other areas of biomedical research? I know you've mentioned it briefly during your presentation about applying it to personalized medicine, but could you expand a little bit further on your thoughts for other potential applications? For example, let's say toxicity testing.
Absolutely. That would really, toxicity testing really is the low hanging fruit for tissue chips in general. We have, especially our pharmaceutical partners, and I'll make a note, but our pharmaceutical partners developed what they call these organotypic manuscripts. And what they did is they said, okay, well, for a cardiac system, what kind of cells do we expect beyond cardiac cells within these models? What assays should be performed? What should be the endpoints? What are the readouts that we need to be able to agree that this is able to answer the question preclinically that we want it to answer? And so those organotypic manuscripts are a great step and a great start to kind of understanding where the pharmaceutical industry is in terms of where these can be used. I do tend to think of tissue chips being used in terms of broader applicability in three major areas. So not just safety and tox testing, disease modeling and efficacy testing, but also personalized medicine. And so when I say personalized medicine, I mean, if you are, say, for example, you're a pregnant individual, you are not allowed to enroll in any clinical trials based on your protected status and that of your fetus's protected status. Okay. But you still most likely need to know which medications you can take safely during pregnancy. What this would allow us to do is take that individual's, fibroblast or blood cells, rederive those into induced pluripotent stem cells, make those into a more mature cell type, and then actually test the therapies on. So really thinking of these tissue chips as patient surrogates. Like, could we run phase 1 clinical trials, which are really just about safety, right? Could we run a phase 1 clinical trial just with tissue chip surrogates, right? And then that way we would not have to take individuals from the population that are what we consider healthy, non-diseased individuals for this phase 1 clinical trial. We wouldn't have to take any of those individuals. There wouldn't be any harm, any potential issues or harm that could happen to those individuals, right? They would just instead be a patient surrogate. And so I kind of think of that as broader applicability. Another answer to that question would be, could we create tissue chip avatars? So could we create you on a chip and then combine basically your cells on a chip with computational data? So you have, all humans have EHR data or electronic health records. Could we combine your electronic health records as well as kind of computational modalities, so genomic profiling, pharmacogenomic profiling? Could we combine all of those with tissue chip avatars to really create you on a chip, to really create a person, a way for you to have a digital kind of copy of yourself to test different drugs and therapeutic modalities on with no risk to you, would be the goal.
Fantastic. Yeah, it's definitely a promising research to look forward to. We did receive a fantastic question quite earlier on during your presentation, and I think it'd be interesting to hear your opinion on this while we're discussing drug discovery. So a viewer has actually asked, what are your thoughts on tissue chips versus organoid models in terms of drug screening?
This is a great question. So for drug screening, and what we're talking about is really like the very early kind of target identification and trying to find hits within preclinical drug development. Organoids are probably a better option at that stage because of how high throughput they are. The fact that we can take organoids and we can put them into 96 well plates or 384 well plates would really, really, really expedite the drug screening that we're capable of doing and really quickly help us understand which drugs are toxic that we can just completely throw out and move on and focus better on candidates that are more efficacious and less toxic, right? And so organoids, I think, are really helpful in terms of throughput. And when I showed that slide on balancing throughput and complexity, that's something that we always kind of have to think about. Taking a really advanced tissue chip model and using it for high throughput screening probably isn't feasible. And we also know that tissue chips in general at this point don't have a very high throughput nature at this point. They just simply do not, based on how complex they are to assemble. It's been very difficult to run them in like 96 and 384 well plates. And so I would say that that would be the benefit of organoid models there. Now, one thing I do want to mention is that per the FDA's recent definition, organoids are considered a microphysiological system. And so I didn't really touch on that in my talk, but the FDA definition of microphysiological systems does include organoids. And so organoids are something that we have focused on within the NIH tissue chip program as well, especially organoids that are vascularized that we can actually perfuse is really kind of the best option in terms of using organoids. And so, yeah, I do think that for drug discovery, especially for high throughput drug screening, we're probably going to need and rely on organoids more than more advanced tissue chip models.
Fantastic. Thank you, Passley. I know we've gone over the time, but I do want to end on just one final question before you go. So looking ahead, what does the future hold for the NIH tissue chip program? Are there specific areas of focus or exciting developments in the horizon that we should be looking into?
Yes, absolutely. Great question. We really our sights now are set on complementary. I included a link to that in the last one of the second to last slide in my presentation. But complementary for us really will be a 5 to 10 year program with a major investment from Common Fund, much higher investment than we've been able to do just through NCATS ourselves. And so I would say wait for those RFAs and those funding opportunities for complementary to be released, because that I think is where we're moving in the field, and that's gonna be very exciting. So I'm really excited, and I think within complementary we will explore this idea of kind of digital twins, this idea of a tissue chip avatar alongside kind of a computational resource.
Fantastic. Thank you so much, Passley. Unfortunately, that is all the time that we have for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. And again, a big thank you to Dr. Passley Hargrove-Grimes for sharing your research with us today. And it has been an absolute pleasure. And to all our viewers, we'll be back soon with the next talk. Thank you.
Thank you so much.